Great. Good afternoon, everybody. Thank you for joining me today in my seminar presentation. I am Paulina, a PhD student at the Namibia University of Science and Technology, known as NAST. Today, I'm going to give you a brief of my PhD work titled The Ecophysiology of Welwitchia mirabilis and Colophospermum mopane, Water Utilization, and Environmental Responses. When we are talking about plant physiology or plant ecophysiology to say, we are basically referring to how do plants interact with their environment and how do they respond to these environmental conditions. It could be water, it could be temperature, it could be light, carbon dioxide, or humidity in order to perform their physiological functions. Throughout this PhD journey, we have a lot of various collaborators, starting with NAST, where I'm registered, and the State University of Arizona, the Swakop-Uranium, which is the mining company that have funded the Welwitchia project, in collaboration with the Gobebeb Research Center, which is now renamed as the Namib Research Institute, as well as the Ongava Research Center, which is the collaborator and the funder for the Mopane project. Starting off with the background to this study, Namibia is one of the driest countries in sub-Saharan Africa, and it's expected to become warmer, with temperature expected to increase by 1 to 3.5 degrees Celsius in summer, while in winter, we are expecting this temperature to rise about up to 4 degrees. So these changes in climate, or to see the weather patterns, they are projected to significantly affect our vegetation. And one of these good examples of the species that may be at risk of climate change is the iconic Welwitchia and mopane. I'm not sure if everybody knows how the Welwitchia plant looks like, or maybe a mopane to say. So on your left-hand side, that's the Welwitchia plant. Right here, this is a male Welwitchia plant, and right close to it is a female Welwitchia plant. And this is the kind of habitat or ecosystem where this
Paulina, sorry, those of us that are online, your slides aren't changing on our screen. It's like the screen's frozen. We can just see your title slide still.
Thanks, John.
Okay, thank you. Let's see if we can fix that. They're changing, should we maybe?
And now?
So what slide are you seeing now?
It's still the same, I'm afraid. Is it the same for you?
Yeah, I still see the title slide with a little blue ring in the middle, which showing that it's hanging somehow. And the blue ring is not on my side, so must be your screen that's showing that.
Could it be the internet connection?
We can hear you and we can see your video. I can see you clearly. Something has changed, but it's not in slide mode. Should it be in slide mode?
We just took it out of slide mode. So,
well, if it works out of slide mode, that's fine.
Yeah, no, I mean, we can still see everything. So, yeah, okay. Sorry to interrupt you.
Well, let's just quickly try this again.
Paulina, I also have an issue. I had an issue as well with my computer trying to use presentator mode instead.
Okay.
So I ended up doing that and just not using the presentator mode. So I'm not sure if it's the video projector or it was on my computer, but it seems that it's.
Are you still only seeing the one slide?
No, it looks good now. Okay, there's some pictures now. All right, we've got.
Okay. And if I change it, you can see the new slide.
Yes.
Oh, excellent.
Okay, thank you so much. So what I was saying is that on your left side, you could see a Welwitchia plant, and on your right, that is the mopane plant. So the Welwitchia plant is a very unique and iconic plant that is found in the Namibian desert, and as well as a part of the northern part of Angola. The plant is a gymnosperm and has been surviving for years in these harsh, hyper-arid environments. And it plays a crucial role in terms of the fact that it provides habitats to the desert organisms, and it also provides fodder to some animals. In terms of the mopane, it's also a drought-tolerant species and is dominant in the northern part of Namibia. And it also provides fodder to both wildlife and the livestock, and as well as it provides habitats to a measure of organisms within the ecosystem. And I also want to point out that majority of us, the people from the north, we highly depend on these species for our livelihood. Now, the question is, what is the purpose of this study? We want to understand what is mopane and Welwitchia water use efficiency. Basically, what you want to know is how efficient is mopane and Welwitchia utilizes the water, and what are the physiological mechanisms do these plants possess in terms of when they are being experienced to water stress. The other point is that we also want to assess this plant's water requirement for future conservation efforts, as this will provide guidance to us, the environmentalists, as well as the policymakers for sustainable management practices. We also want to confirm the Welwitchia water usage. Currently in the literature, it's indicated that Welwitchia uses about 1 liter per square meter per day. That's the transpiration rate currently in the literature. We think that this is a ridiculous amount of water this plant is losing, provided it lives in such a hyper and such a dry ecosystem. So this map shows the distribution of the Welwitchia plant across the Namib. But what I want to point out is our study areas. The first one is the Hope Mine right there, which is close to the Gobabeb Research Center, our collaborator for the Welwitchia project, and as well as the Welwitchia Plain. This population is close to the Ecophysiology, which is a mining company that funded the Welwitchia project, and we also sampled in the Kunene population. For the purpose of this presentation, I have narrowed down my studies objectives into 2. The first one being to measure stomatal conductance over diurnal cycle. This objective is specifically for Welwitchia. So what we have done is that we carried out 72 hours experiment over the 3 populations to determine the water use of the Welwitchia plant. The second objective is the integration for both Welwitchia as well as the mopane, which is to assess short to medium-term response and seasonal variation to moisture. This is now in respect to various plant health indicators, which is photosynthesis, stomatal conductance, transpiration, leaf water content, chlorophyll fluorescence, as well as leaf phenology. I'm sure that not all of us are familiar with these terms, so I want to unpack each of these so that we can understand what they mean. Starting off with photosynthesis. What is photosynthesis? Photosynthesis is the process whereby plants convert light energy in the presence of chlorophyll to produce carbon dioxide, or carbohydrate, or to say glucose, and oxygen. But how do plants do this? Let's start off with the simple leaf, basic anatomic structure of the leaf. On the top layer of the leaf, you would find what you call cuticles. The cuticles, these are the wax transparent layer that you find on the plant leaf. And what they do is that they prevent excess water loss from the leaf surface. Below the cuticle layer is what you call the epidermis. The epidermis, I call it the skin of the plant. Just like us humans, we have the skin that protects our internal organs from the outside environment. The epidermis of the plant plays a similar role. Below the epidermis is what you call the mesophyll cells, or these star green cells that you see there. That's what you call a mesophyll cell. And these mesophyll cells, this is where exactly now photosynthesis takes place inside the plant. The mesophyll cells is then subdivided into two. We have the parasite mesophyll cells, which is close to the epidermis layer. And then we have the spongy mesophyll cells, which is below the leaf surface. So the parasite mesophyll cells, what they do is that, or to say that their role, it's to transfer the light energy that is absorbed from the sun, and they transfer that energy into the spongy mesophyll cells right here. So the spongy mesophyll cells, this is a major site where photosynthesis takes place inside the plant. So within these mesophyll cells, you would find structures called chloroplast. And inside this chloroplast structures is now what we find this molecule called chlorophyll. It's a green pigment molecule inside the plant leaves that is now responsible for the absorption of light energy. So what happens is now, when chlorophyll absorbs the light energy from the sun, the light energy helps to convert water that is being absorbed from the soil and carbon dioxide that is being uptaken from the atmosphere to produce glucose, which is the energy that plants use to carry out all their functions for growth, as well as oxygen that we human beings breathe in, that you basically need to survive. Moving on, it's the stomatal conductance. What is stomatal conductance? This is the rate at which the stomatas on the plant leaves open and close to regulate or facilitate gas exchange. I want to take you back to the basic structure of the leaf. If you could see here below the leaf is what you call the stoma. The stoma, it's a singular, and in plural, we call it stomata, which is a bunch of small holes, or to say opening on the plant leaf. So this stoma, it's now the small holes or tiny small holes that we can't able to see with naked eyes. And this is what is responsible to facilitate gas exchange. When I'm talking about gas exchange, I'm basically saying that the uptake of carbon dioxide from the atmosphere, as well as the evaporation of water vapor within from the mesophyll cells inside the plant. So that's what we call now stomatal conductance. And if you could see here, this is also the stomata. And if you could see on each side of the stomata, or to see the pore, you will find these cells on each side, which is known as the guard cells. And these are the cells that are actually responsible for the opening and closing of these stomata, or to say the stoma. I'm not sure if you can, I have put up a well-zoomed picture there of how the stomatas look like, but let's move forward. Transpiration, it's basically a loss of water from the plant leaves. And water use efficiency. Water use efficiency is a measure of how efficiently plants use water to carry out photosynthesis. We also spoken about one of the plant health indicator, chlorophyll florensis. Chlorophyll florensis is a measure of how efficiently the plants use the absorbed light from sunlight to carry out photosynthesis activities. So what happens is that, as I have mentioned earlier, that the chlorophyll molecule is the one that is responsible for absorbing light from the sun. So sometimes not all the absorbed sunlight, or to say light, is being used for photosynthesis. For a healthy plant with active photosynthetic structures, or to say machinery, in most cases it only uses about 82, or to say on average 80% of the light for photosynthesis, and the excess light is actually being released back into the atmosphere, and this is what we call florensis, and this is where the term chlorophyll florensis came about. So from chlorophyll florensis, it's a very good indicator for us scientists to measure or to monitor plant health, and also to see whether the plants are healthy or stressed. When a plant is stressed or unhealthy, you would expect more light, or to say high light intensity, being released back in the atmosphere. So what are the research questions that we are asking ourselves? So for the mopane project, I have one overall research question, which is, what are the effects of the seasonal changes in the indicators that I have mentioned earlier, which is photosynthesis, transpiration, stomatal conductance, leaf water content, chlorophyll florensis, as well as the leaf phenology. As for Welwitchia, I have noted down a multiple of research questions. The first one we want to know, is there a variation in the stomatal conductance across different geographical areas? And these geographical areas, I'm talking about the three populations that I have spoken or indicated in the study map. The second one is, what is the variation of the photosynthesis in photosynthesis, conductance, transpiration rate, chlorophyll florensis before and after the watering event? So this was a watering experiment that we actually conducted, or we actually did on the Welwitchia. And the third one is, how does stomatal conductance affect the rate of photosynthesis as well as transpiration? The last research question, we want to know whether photosynthesis and transpiration varies across the leaf age of the Welwitchia. So as you can see here, the Welwitchia plant has, as I said, it has 2 leaves, or maybe I haven't mentioned earlier, it has basically 2 leaves, and these leaves, they grow persistently across its life cycle. So what we did is that we measured different leaf ages, to say close to the stem, across the leaf gradient up to the leaf tip, to see if there's any variations within different leaf ages of the Welwitchia. So when it comes to the methods for photosynthesis, stomatal conductance, transpiration, as well as water use efficiency, I use the LICO, which is a portable photosynthesis instrument. And I have also mentioned that one of the objectives is to determine the stomatal conductance of the Welwitchia. What we did is that we have used this instrument known as the leaf barometer. As for the chlorophyll florensis, this is the instrument that we use, known as the HandyPA. Furthermore, let's look at additional measurements that we are currently busy with the mopane. We are measuring the leaf health, or to say we are monitoring the leaf health to determine the phenological changes within the mopane leaves. And here I'm using a grading system ranging from 100% green leaves to 100% brown leaves. I'm also measuring the gravimetric soil water content, leaf water content, as well as the weather data just next to the experimental site. For the protocols in terms of the experimental design, we have a total of 16 plants. 8 of those, they are being watered, and 8 they are not being watered. Basically, it's unwatered plants. So each of the watered plants, they are being watered every second week for 2 hours with a hose pipe. And throughout this experiment, I have considered to measure the green leaves for both the unwatered as well as the watered plants. The yellow leaves being measured on the unwatered plants. However, because I couldn't find yellow leaves on the watered plants, I opted to measure the green spotted leaves. And this experiment will be running through the dry season, and we will carry on during rainy season. So in today's seminar presentation, I'm going to share with you my preliminary results from the ongoing research on the mopane project. And first, we'll be starting off with the stomatal conductance, the responses of stomatal conductance over time. So what we did is that we have measured these plants just before 9 until 5 in the afternoon. And you could see the first two scatter graphs. These graphs shows the results of unwatered plants. The first graph is for the unwatered plant, and the second graph is for the watered plants. And this is the error bar chart just showing the distribution of the results. So just to explain to elaborate as well, on your y-axis, that's the stomatal conductance donated as GS, and it's being measured in the unit of moles per meter square per second. And on your x-axis, that's the time of the day. The gray triangle shows, or to say that it represents the green leaves in both the graphs. And on the unwatered plants, the circles represent the yellow leaves, while on the watered plants, it represents the black spotted leaves. And if we are to move now to the bar chart, the gray bars represent the green leaves for both watered and unwatered. And the white bars for the unwatered, it's for the yellow leaves, while for the watered plants, it's for the black spotted leaves. So let's dig deep what we just find out. So what we found out is that the stomatal conductance, it showed a nice wall diurnal pattern, where stomatal conductance was high in the morning and gradually declines throughout the day. This is a pattern throughout that we have for both watered and unwatered plants. And the declines, the gradual declines that we see in the afternoon, it was more pronounced in the unwatered plants compared to watered plants. And if we move to the bar chart, we can see the distribution here clearly, showing that the watered plant has much higher conductance compared to the unwatered plants. You may ask yourself, but why such a decline? You know, from the morning, and then all of a sudden we have such a peak decline over there. The reason why we have such a kind of a decline is because plants respond differently to these climatic conditions. So you would find that midday, when the temperature, or to say the light intensity, start increasing, the plants also start responding differently. Temperature has a bad effect on the plant health. So when the moment that the plants start experiencing high temperature, what they do is that they create a negative gradient inside the plant leaves. So the plant leaves start having such a negative gradient, which forces the plant, the temperature, what it does is that it forces the plants to lose a lot of water at a faster rate. So with that loss of water, what it does is that it now forces the plant to absorb more water from the soil. At the moment that the plant fail to meet this transpiration demand, what the plants do is that they start closing their stomata. They'll be like, hey, there is a lot of stress here that we can't able to maintain. So the plants, they start closing their stomatas. Hence, we have a decline here from the morning and throughout the day. And what I also want to point out is that the observation that is striking also in these results is for the yellow leaves. We could see that the stomatal conductance of the yellow leaves is quite lower compared to the green leaves as well as the spotted leaves. And the reason why the yellow leaves has lower stomatal conductance, it's because when you look at the yellow leaves, it represents senescence, which is the aging of the leaf. Number two, yellow leaves have also a lower chlorophyll content, which is responsible to absorb light energy. And most of the structures in the yellow leaves are completely destroyed. That includes now the photosynthetic structures as well as the enzyme and also proteins that are responsible for the biochemical reactions. So all these factors, they affect the functionality of the stomatas. Hence, we have a lower stomatal conductance in the yellow leaves. As for photosynthesis, we have more or less a diurnal pattern as that of the stomatal conductance. And this is because photosynthesis really depends on the functionality of the stomatas inside the plant leaves. So when the stomatas, they are open, that means that the plant is able to absorb carbon dioxide from the atmosphere, which is very essential for the photosynthetic activities. So photosynthesis was also high in the morning, as you can see here, for both watered and unwatered plants, and gradually start declining throughout the day. And if we could see also the yellow leaves, we got the same kind of pattern as we have explained earlier, that it was lower, close to zero. That is the same pattern that we got in photosynthesis, and this is basically because these tomatos of the yellow leaves were either partially open or they were completely closed. And that leads to the reduction of photosynthesis activities as well. And overview, we can see that photosynthesis was also high in the water plants compared to the unwatered plants. As for transpiration, we could see that transpiration was lower in the unwatered plants compared to watered plants, and that tells you, or that suggests, that there is limited water availability in the soil. And but what is also striking here is the response of yellow leaves throughout the day. For photosynthesis and stomatal conductance, those values were quite close to zero. But in this case, these yellow leaves, most of this little available water, it was being transpired. that tells you that these leaves, they're not water efficient. We also spoken about water use efficiency as the measure of how efficient the plant uses water for photosynthesis, as well as chlorophyll fluorescence, which is the measure of how efficient the plants use light energy to perform photosynthesis. So what we found out is that despite the differences in the water availability for unwatered plants and watered plants, these plants manage to effectively use the water for photosynthesis. As you could see that the rate is between 0 and 200. However, this is different from the yellow leaves. The yellow leaves, we could see that the water efficiency was quite much lower. This tells you that the little available water in the yellow leaves were being transpired instead of being used for photosynthesis. This is the kind or same pattern that we have also observed in the chlorophyll fluorescence. And we could see that both the green leaves of unwatered plants and also watered plants, including the spotted leaves, about 70% to 80% of the light that is being absorbed by the plant is being used for photosynthesis. However, if you look at the yellow leaves of the unwatered plant, this plant was actually re-emitting much of the light that was being absorbed by the chlorophyll, and which leads to the lower efficiency of light energy. So we could see how different plants respond to different climatic conditions and how different leaf types also respond differently. So based on the preliminary results that I have just presented, we can see how it's important to maintain the sufficient water supplies to ensure optimum plant performance as well as growth. Therefore, understanding plant physiological mechanisms is crucial for enhancing sustainable management practices. Thank you. That was the end of my presentation today. And it's time for the questions, recommendations, opinions, etc.